All right, our next speaker is Xiaohan Tian from Beijing Normal University, and he will speak to us on the topic of spontaneous brain regional dynamics contribute to generalizable brain behavior associations. Let's welcome.
Thank you. Good morning, everyone. I'm Xiaohan from Beijing Normal University, and today I'd like to share with you our latest work about spontaneous brain regional dynamics contribute to generalizable brain and behavior associations. I guess many of us would agree a big reason we study resting state brain activity is to understand the individual differences. This is why people think, feel, and behave differently. And this line of research is now often called by brain-wide association studies, or BWAS. And a typical starting point in BWAS is to define features that capture meaningful patterns of brain activity. And almost by default, we turn to resting state functional connectivity, or RSFC. It tracks how signals from different brain regions correlated over time and have played a central role in many landmark BWAS research. But what if we take a step back? Activity from individual regions may reflect a more basic unit of brain function, yet is often overlooked. We here believe it deserves more attention. For one, it can help us connect what's happening across levels, just from the single neuron to distributed networks. And just as important, a region's activity over time tells about brain's flexibility, something that is essential for adaptive behavior. So that's why we here focused on resting state regional dynamics, or we call it RSRD. Even within a single region, the brain dynamics can be surprisingly rich, with patterns like variability, complexity, and other various temporal structures. But most studies still stick to just one or two hand-picked features. This situation is a bit like the blind man and the elephant. That is, each one touches a part of elephant, but no one tells the whole picture. So to really unlock the RSRD's potential in BWAS, we need a more comprehensive perspectives. And of course, bringing new brain features into BWAS raises some key challenges, such as robustness of individual specificity, their behavior relevance, or can they be generalized across a broader application. So taken together, our motivation here was simple, to explore the potential of RSRD in BWAS framework. And to unpack this, we focused on 4 key questions, which I will walk through one by one. And we here applied 2 key resources to carry out this study. First, to comprehensively capture regional dynamics, we applied HCTSA, a toolbox that runs thousands of temporal features on each region's activity. And this gives us a feature-level profile matrix of each one's spontaneous brain activity. And we leveraged data from 4 open datasets, 2 of them from the Human Connectome project, the Young Adult and the Development, and also Midnight Scan Club and the UK Biobank, each differ in age groups and the sky designs. First, for the first question, how do we build RSRD profiles at the individual level? We here focused on the reliability, which is essential for individual brain profiling and meaningful brain behavior mapping. And by calculating the interclass correlation coefficient ICC for all RSRD features output from the HCTSA, we identified 44 reliable features, each showing an average ICC above 0.5 across the whole brain. And these features span 4 dynamic dimensions and provide the basis of our following analysis. Since feature reliability can be affected by various factors, we tested the stability on the different setups. And we first validated within the HCPYA with varying sample size, the motion level, and the number of scan runs. Each dot here shows the feature's ICC on the different condition. And as you can see, the ICC values for the RSRD feature are highly correlated across conditions. And the refined features identified in the HCPYA group 1. here labeled in green, was consistently ranked as the top. And this high feature reliability and their agreement was consistently replicated across the independent datasets, the MSC and the HCPD. So to answer the first question, we find ISRD reliably capture individual differences across contexts, thereby enabling the robust individual level brand profiling. Next, we ask whether RSRD captures meaningful behavior associations. And to test this, we applied a canonical correlation analysis to link the refined RSRD features with a set of brand behavior items from the independent HCP-YA participants. And we got two significant brand behavior associations. The first mode was primarily linked to the recent smoking and alcohol use, so we called it a substance use mode. And the second was mostly related to the cognitive ability, so we refer to it as the cognition mode. And we find that RSRD contributed differently to each mode. For the substance use mode, the key contribution came from the sensory motor network and the subcortical regions, and with dynamics mainly reflecting the temporal dependencies, such as correlation. For the cognition mode, the default motor network and the cerebellum contributed the most, and with a greater influence from the stochastic dynamics, such as the random walk. So to answer our second question, we find RSRD supports two different brand behavior associations, and each with a clear definition from the both behavior and the brand aspects. After identifying the clear brand behavior associations in HCP-YA, we further wonder whether they can be generalized across different datasets. So we introduced the two different datasets. The HCPD covered children and teenagers, and the UK Biobank included the middle-aged to older adults. And we here use the same RSRD features and the HCP-YA trained model to generate the model-specific latent scores, and then test whether those scores still capture the relevant behaviors in the new cohorts. And in the substance use mode, we find that the younger participant from the HCPD showed links to the M-rated gaming, while in adults and the older adults, the same brand pattern related to the smoking and alcohol use. But in contrast, the cognition mode was consistently linked to the fluent intelligence, even though the task across course was entirely different. And interestingly, this age-specific pattern in substance use mode is consistent with previous studies showing that playing violent games during adolescence can predict the later addictive behaviors. So taken together, despite major differences between the cohorts, we found that the brand behavior associations can be generalized across a broad population, but the substance-related behavior may underline a developmental shift. Finally, what does RSRD offer new perspective for the BVAS? We believe its key contribution is giving the equal attention to both the spatial and temporal aspects of spontaneous brand. And by capturing dynamics across multiple temporal dimensions, we find that different dynamic patterns may relate to different behavior. And specifically, the cognition was most associated with the random walk-like dynamics, and which is often used in modeling cognitive process like decision-making. And in contrast, the substance use was tied to autocorrelation, reflecting how much the current brain activity depends on the past. And we further found that those behavior-linked dynamics show the consistent spatial patterns across datasets. So for each mode, we can tell the RSRD provides a generalizable triangle connecting where and how racing brand supports different behavior. And to sum up, we think RSRD highlights the role of temporal diversity of spontaneous brain, and thus give us a new way to understand how spontaneous brain to support different behavior. And in summary, we here use the resting state regional dynamics RSRD to build a reliable and multidimensional framework for individualized brain function, and revealed two different brand behavior associations, one for substance use and one for cognition. And we find that they can both be generalized across datasets. So taken together, we believe RSRD offers strong potential for advanced BLAS, and you can find associated details in our GitHub project. And I would like all my supportive mentors, collaborators, also the open data community make this work possible. And you can find me at the poster season at 1682. Thanks so much for listening.
Thank you, Xiaotian, for the very nice talk. There are a couple of questions from the audience. So the first one is, could the increase in synergy and redundancy be explained just by global increase in total amount of information? Global increase in the total amount of information.
That means the data sets or the features. Because I'm very not sure about this question specific aspect. If I understand it correctly, it maybe the effect size can be amounted by the sample size?
Yes, maybe, I guess.
Okay, maybe. That is a very important question. I think, but in our study, we find the sample size or the feature size may be a little bit influenced for the study, such as the UK World Bank included 20,000 people. But the HCPD is a relatively small group and still capture the same conclusion. So I don't think the mountain of informations may affect our results.
Okay, thank you.
Thank you.
Another question, how can you disentangle the dynamic properties of a region from the inference of connected regions?
Okay, that's also a good question. We think the regional dynamics maybe play a complementary role to complement the connectivity patterns as it attracts a different type of information. And maybe in the future, we can combine those two information to further advance in BOS.
Okay, maybe very quick answer to this one. Would DCC run across data sets or separately for each data sets?
It's separate for data sets.
All right, thank you. Thank you again for the speech.